Go ahead and get started. I'd like to welcome everybody here today. My name is John Carr. I'm a history professor here at Kingwood, and this is the third speaker this semester in our faculty speaker series. This is our final speaker in this series for the semester, and we'd like to welcome you all here. My father-in-law is 80 years old, and he just turned 80 a couple weeks ago, and he thinks a lot about the past now. I hear him talking more and more about his childhood or about his experiences at NASA and that sort of thing. Well, he recently got a DNA test, and the reason he got a DNA test is he wanted to know about his ancestry. That's very interesting to him. He wanted to, actually, what he wanted to know is if he was part Irish. Turned out he was, he thinks, okay? But maybe not as much Irish as he hoped. So, the question of DNA and ancestry, this is something that fascinates Americans. I know my father was very involved in genealogy before DNA testing. But what Dr. Szymanski is going to talk about today is very new. It's a new science, if you will, and he's going to maybe throw some light on whether this is actually a science at all, or to what extent it really is. So, without any further ado, he's got about 30 to 35 minutes and he's going to talk, maybe a little more, and then he wants to take questions and answers from people. Dr. Brian Szymanski.
Thank you. And could everybody hear me? Because I have a very loud voice. I just want to make sure I don't yell over the microphones. Is this okay? Okay, good. And I always sound weird over a microphone. So just a little what we're going to do today is I spend enough time boring my students with biology and stuff like that, so I'm not going to go into too much detail about the biology of the genetics. I'm here to build a convincing case of if you do this type of stuff, because how many of you are interested in doing a DNA genealogy? Because especially since the price goes down. How many of you would when the price goes down and you have money to do it? I should ask that for students. Good, very good. Because I've given this talk almost, I think, 100 times now over the past 20 years. That's how long this has been around. And one thing you should also do before you hear a speaker is ask, what's their credibility? Because I am a practicing scientist, not just a teacher. So I'm still involved in particular types of research related to my graduate work. And so why am I talking about this in the first place? Because when I was in college, I think Watson and Crick were still developing the structure of DNA. And I'm serious about that. We didn't know what genes were about. We didn't know a lot of stuff. The genetics, I don't know how they spread it out over 16 weeks because we knew very little information. And it was coming out as I was learning it. And it wasn't to graduate school that I finally did the technology you see here, which literally required intensive training. It took almost a whole semester to sequence a tiny little piece of DNA called the insulin gene, which was well established back then. We just did it to see if we got it right. I remember taking like 10 weeks to do that, and it was such a specialized skill. And now I do workshops for little kids teaching 9-year-olds how to sequence a piece of DNA in no time, which is really kind of fun. We call real time. And that kind of bothers me. So it's like, they're doing what I required a graduate degree for. So whatever, so the technology's gotten cheaper, it's gotten faster, it's gotten simpler, and the science is excellent, but the commercialization I'm gonna talk about today is kind of stinky. And my run-in with all of this, particularly the genealogy stuff and the disease stuff, because now, and this is my laser pointer, because the laser doesn't work on that board. So now we're learning one company up here, 23andMe, has gotten permission, FDA approval, Food and Drug Administration approval, to use their data for clinical diagnosis. That means it has legitimacy of looking at disease, which I don't want to get into too much today, because that's totally different genetics than looking at the genealogy. I mean, it's somewhat related. It's correlated, but not directly related to what we're going to look at today. So so basically what happened was in 1989, and I don't know how I fell into this, but I heard about what was called the first human genome project taking place. And this was the first attempt to sequence all human DNA. And they were expecting a 15-year project, which ended up taking less time because of some people that were looking at the commercialization and sped it up a little. But I remember being at the first conference ever meeting Watson and Crick and all these biggies. And I was a brand new professor at Oklahoma. My only experience with DNA was in industry. Actually, I was an industrial chemist developing genetic products. But it was a growing field. I was just barely learning about it and making the products for what you see today. Matter of fact, some of the kits we made are still used today. So I was not really into all of this sequencing stuff because it was a horror when I was in graduate school to do this. But eventually I got thrown into this controversy of we're going to sequence a full human DNA, and a sociology colleague of mine, he was from the University of Las Vegas, Nevada, said, let's get together and survey literally the United States, Canada, and Mexico. And I'm like, oh, okay, that sounds easy. And understand, this was in 1989. We didn't have the internet to do that stuff. So we used this stuff called real mail and real surveys we mailed out, and it cost a lot of money. It wasn't just a blink of a finger. So we surveyed all different people, different socioeconomics classes, and asked if you had this available, this technology, to understand your genes, your lineage, your handicaps, what would you want? And we presented this paper. It was a beautiful paper. We had it broken down to religion, socioeconomic class, race, and we found some very ugly information. And that related to something called eugenics, the human betterment, which is funny because we were proposing that to the scientific community, the scientists and the sociologists. And basically, after our session, Dr. Watson, who's of Watson and Crick fame, came up and said, those guys I gave them one paper, what a bunch of hokey. So I was never insulted before by such a famous person. And he said, this is garbage. No one will ever want to abuse genetic information. 2 years later, insurance companies were looking at using it to determine whether they want to insure you or not, or how much to charge. And doctors were requesting it, and it just boomed and boomed. We were at a conference in 1992 at the University of Houston. That's right before I got here. And I thought in the winter, Houston was kind of cold, so I brought a winter coat along, knowing little it would be like 80 degrees, whatever. I still wore the coat just out of arrogance. Okay, but, and we were already setting the tone for the ethics of all of this. So most of my interest in this is not so much as a scientist, but as a person that's interested in how do you use genetic information. And that's where all these companies start to blossom. Genome 20, which used to be called the Genographic Project, they're the founders of this. And they were looking at pure research and interest. And they developed the databases for Ancestry.com, MyHeritage, these are the biggies, and 23andMe. And guys, I have no vested interest in any of these companies, except 23andMe gave me some type of award, because I gave 100 talks like this. And they gave me an award, I think it's like Miss Congeniality for promoting genetics, but I'm not going to promote their product. What I want to give you is the truth behind what is this information, a little about the biology so you know what you get. Because I've had people come up to me and said, hey, I'm part Neanderthal. I'm like, okay, yeah, and we're going to look at what that means in a minute. And I tend to disagree with a lot of scientific community on where the Neanderthal came from. But to begin with, guys, why do we do DNA testing for genealogy? Because normal genealogy basically sucks. And I know that from coming from an orphan family. I came from a refugee family where most of my relatives are dead, so family reunions are not hard to arrange. I'm serious, my last name is it. That's it, we're all related. So it looks at relationships, obviously. And it looks at if two people descend from the same ancestor, obviously. If we're related to others with your last name, which we know we're not because it was given to us. I don't even know what our last name was. And I don't know why they didn't call me Smith. Why the heck do we have to get a Belarusian name like that? So these ways are, the traditional genealogy with names and talking to relatives we're finding out doesn't work. And we call that sometimes the brick wall. So you find wrong family members, you find that your last name was not the same, you find out that you have relatives that are not really related and stuff like that and all of that, and it's really difficult and time consuming. And I wouldn't trust many of the traditional lineages that we do. So what the genetics provide us with is actual data. DNA is so intimate, but we're gonna see it's intimate in different ways. You don't treat your friends the same way you would treat a spouse, significant other, whatever. I hope, unless you're just, whatever, I don't care. Okay, so whatever, I kind of keep a distance to certain people. So, so when we get, so this brick wall leads us to this thought that DNA is so intimate, It's been used in many settings to do look at lineage and self-identification and paternity and all this type of stuff. Why not for genealogy? And that's what National Geographic was looking at with the Genographic Project. They hired a PhD anthropologist who also had, I think he had 2 PhDs, he collected them, and one in molecular biology, obviously, and his goal was to find out, do we have a lineage we can trace through DNA? Because DNA is not always reliable. In the past, we've used other molecules to trace, and I'll talk about that as we go along. So this is one way to look at genealogy, and we're looking at it as the easy fix, because DNA is measurable, we can put it up to statistics, we know it's absolute, it's reliable, because you can't lie about it, and if you're adopted, you find out real quickly, unlike having to wait years and people lying to you and things like that, and it tells you true distinctions. And this was a journey for me with National Geographic, because I petitioned to have mine done. Because my dad was an orphan, his part of the world that he came from was known to have a large population of minorities, which he belonged to, and we knew nothing about him. We didn't even know what day he was born. We didn't even know where he was born. So this to me was a trip for me to understand my dad, and he never talked about the past. Refugees usually don't, because it's too bad a story. And my mom's family, we kind of knew about a little, but again, they came from a part of the world which didn't keep records. So, so I did her family too, just to get an idea of who we are. But I understood the limitations of this. I was looking for a different picture than most of you might be looking for out of the 23andMe. And again, we could use DNA to look at genetic risk. And that to me is scary when you start looking into that. But yes, we can determine genetic risk at the same time we're looking at your lineage. It's a very simple process, costs a few extra bucks, and people are willing to shell it out. So just some biology 101. And I'm going to keep this very simple. So humans have 46 chromosomes. You get 23 from your mom, 23 from your dad, you know, whatever. So you're kind of like this little hybrid creature. And think of chromosomes as basically like books. And I brought some books. And guys, I did publish books on this. Those are actually ones I wrote. Okay, so whatever. So it's like little books. And I happen to bring genetics books. I think that's kind of funny. One with the killer tomato and one with some researchers on it. And the books you inherit are unique to your parents, just like these books are. But these books we're going to eventually call homologous chromosomes. How many of you remember that and then forgot it from biology? Okay, how many of you remember it, period? Okay, how many of you even slept through that? Okay, yeah, okay, at least you're honest. So we have these things called homologous chromosomes, which means this has similar information to this. They're not quite identical, and they have a little uniqueness that we can trace through our families. So that's what's kind of neat. We both carry mom and dad's DNA. So basically, you are not you. You are a combination of your parents. So if you have anyone to blame about your behavior, blame them. Okay, that's what I love about genetics. I'm always telling my mom that. You did it to me. Okay, so we have these chromosomes. Okay, and I think of chromosomes as socks. I brought some socks. So again, so you have, you know, one from your mom, one from your dad. They're very similarly structured, but these little white and blue bands might have a little variation. And most of this variation you see in your facial features, in your body anatomy, in your body chemistry. Like how do you process alcohol or not, and things like that. How do I respond to food? Do I have allergies or not? So here's all these little genes. And then we have my unique ones, the X and the Y. And the Y is what we call a truncated chromosome. I mean, it's missing a part. And some people even call it defective, which I don't kind of like that, because I'm a carrier of a Y. Okay, so we have this X and this Y. And what's neat about them, unlike other chromosomes, we're going to see that they are what we call true inheritors. So we have these things called autosomes, and I'm going to show you pictures, which we can test for lineage. And then we could also test these for lineage too, but there's some problems with doing that. And I'm hoping to bring that out. So when you do get this test done, you know what's going on. So, oh my gosh, a cell diagram, I know he'd do it. So when we look at lineage, guys, they're collecting some cells from your body, and there's different ways we can do it. I actually like doing biopsies where I dig through your skin and muscle, but that's not ethical, you know, treatment of a person. So usually what we do is we use safe tissues. In the old days, we used to do blood, and now we do spit. Okay, and I'll talk about that, because the problem with spit is like right now, my spit has pizza genes in it, and stuff like that, and also bacteria, and I don't want to be put into their lineage. So what we're going to focus on is what we call the genomic DNA. That means the DNA that you actually pass down as an organism breeding and having children, whatever. And then we're going to look at these things called mitochondria, which are actually little bacteria-type creatures that travel down, at least in humans, in the female lineage. In other organisms, it varies. So these are little creatures that live in you that came from your mom's egg. Dad just contributes DNA. Sorry, it's a hit and run. Just drops his DNA off, leaves it behind. What we know in the literature is actually mom only uses a small percentage of dad's anyway because she doesn't trust his sperm. And that also affects your, unfortunately, that also affects your appearance. We'll learn when we start looking at genealogy, trying to determine what do we look like. So here we go. So this is your DNA. This is your DNA. And this is, and when we look at chromosomes, actually, guys, this is your duplicated DNA. So this is your DNA kind of like, you know, this kind of duplicated, because this is the only way you can study it. But when we look at DNA, we got to separate the different types. So we separate 22 all the way to 1, and then we have the X and the Y. And so guys, so you're inheriting. When you're being made, I should say, okay, your parents' cells have to determine, do I give my kid this sock or that sock? So this sock I might have gotten from my dad, this one from my mom. And the cell doesn't actually do that. It doesn't say, hey man, what do I not have? Okay, I mean, we can do that in the lab for you, I'm sure. But this just occurs randomly. So I might take this chromosome, let's say this is chromosome 21, that's kind of an important one, and I'm gonna just play this game where I'm gonna make a baby, throw that down, and that's the one I contribute to my sperm. I'm not gonna make an egg for you today. Okay, this let's say is chromosome 3, so now I have 2 chromosome 3s and even labeled paternal and maternal. I have an M and a P in there, boy am I anal. Okay, so whatever. So what I'm gonna do is drop down dads, I could have done moms, I do this several times, and I got a kid that I'm sharing with somebody else. So part of half of me, my parents, a mixture of it, goes into the kid. And this is a difficult thing to trace. When you're trying to trace lineage with these, the problem is we don't know where they're going. And your brothers and sisters might be different pieces of your mom and dad. That's the problem. You might have DNA that heavily, comes from your dad's dad. Actually, you inherit your grandparents' DNA. This is getting more confusing now. So you may be more of one grandparent than the other, the maternal or the paternal. So this DNA is a kind of an unpredictable mixture. 1 to 22, what we call these autosomes. And what's bad about autosomes too is they're sloppy. That means when you have autosomes together, moms and dads, they kind of get together. And some of you know that as chiasm formation, crossing over. They'll swap parts. So now this walks away with parts of mom and dad, and this one with parts of mom and dad. It's no more pure. So these chromosomes, we learn over time, lose their purity. And it makes it very difficult to trace lineage, because you don't know what you're getting, and you can end up actually with no DNA from one of your family lineages, and a lot from another. And it statistically could be disproportionate. That's legal in statistics. So what we focus on usually is 2 pieces of DNA, and unfortunately not the X. This is one part where females don't rule, because the X, when you make a boy, you know that has to be mom. If mom gave you a Y, you're in trouble. But this could be, well, not really, I mean, whatever. But so this could be either, this X could be either your mom's dad's X or your mom's dad's. I should say let me start that over again. That could be your mom's dad's or mother's or whatever, or your father's. It has to be his mother's, whatever. The Y is kind of neat, because every male passes his Y to his male ad infinitum, all the way to the beginning of time. Females don't do that, because the X eventually, you can get X's that are lost, because when you have 2 X's, it acts like an autosome. You don't know which one you're getting. The Y has to be purely passed down from a male. But we can trace these back a long time. So this is called traceable DNA. This is called a mess and hell to track. That's actually the technical term. So there's something else we do now too, is we learned that mitochondria pass down from female lineage purely. So you can tell not just your mom from you, and males and females can do this because we all get mom's mitochondria. Nobody gets dad's. His ends up getting sucked alive and decaying and whatever, once it fertilizes the egg. So we can trace mitochondria just like sperm. the Y in the sperm, going all the way back in time to, and we're going to see what we mean by all the way back in time. So the typical genealogy that you would get from, let's say, Genographic Project, and most of these places, they first look at your mitochondrial DNA, which looks at your mom's inheritance, and then your dad's for the Y. If you're a female, oops, you better find a male relative. Okay, that's the problem, because you don't have a Y. Okay, and we look for these things called polymorphisms. That means changes in the DNA over time. And there's a big debate on which ones to use. We have ones that are called simple mutations, SNPs, ones that are called repeat mutations, which are called STRs sometimes, or hyper-STRs. And what that means, guys, when you have DNA, the DNA that you have right now in your body, and I'm calling that a mutation, can be modified by what's called these mutations. And these mutations we can trace in every gene. Okay, the problem with these mutations is that they can be subject to environmental change, what we call natural selection, and that could skew this. Okay, so what we look for is DNA that is what we call stable and also has a regulated clock, because I can put these clips on very quickly or very slowly over time. So these clips can occur in one generation, or I can have this DNA pass on 10 generations and add a clip. So these are what we call our genetic markers, and sometimes our genetic chronological markers. And I will find that unique families have unique places where they put these mutations. It has to do with the structure of the DNA. So, but we have to look at the right DNA is the question. So when we go back to the chromosomes and we start looking at this, we know from the literature is that Y chromosomes and mitochondria have all sorts of types of DNA. Stuff that's called conserved DNA. That means it doesn't mutate much over time, and it's very stable. The problem with the conserved is I can easily find out that you're related to an earthworm very closely, and that's a problem, because that doesn't mean I can go out and get a marriage license and now marry an earthworm. I mean, in Germany, I think you can do this. Sorry, Thilo, but you know they got a little liberal with that for a while. Okay, so whatever. In Hungary, we don't allow anything. Okay, so whatever. So seriously, so you can do that. And guys, even when, and then we can look at what are called sometimes structural genes. That means the genes that make you what you are. You'll find out that you're more closer to a cow than a person sitting next to you. And that's true for chimps. We found out that our relationship with chimps is so close, sometimes humans are more related to a chimp than a human as far as these genes go. And that's kind of a little silly too. You don't care about that type of lineage. And that's what these databases are not looking for. They're looking for another set of genes that tend to be what we call a little less conserved. That means more mutations, but are also not related to evolution. And we find this in what's called junk DNA. That means DNA we thought does nothing, it's stable, it's supposedly not affected by your development, by the environment. They just sit there, and over time, you pass them down generation to generation, adding what? These little mutations in very specific spots and sometimes at very specific times. And we've known about these for a long period of time. People that study animal migration have been doing this for a long time. And the way they do this is they look at what's called, they think, the ancestral family of, let's say, mice. I actually watched a study on mice that lived in New Mexico, whatever. And we knew they were an old population. And what happened was, as the population got big, they moved, and we're able to track the mutations as they moved and stuff like that. And we can tell how long it took to move based on these little chronology genes. So what's neat about DNA is it timestamps you, and it tells you who you came from. But you have to pick the right DNA, because you don't care that you're related to a fly. You don't care how distant you are from a fly. You don't care if you're distant to a cow or not. What we're looking at is distance to other humans, and that becomes a difficult thing. So to move along, these mutations, again, are just slight changes in DNA. This one favors what we call SNPs, simple ones, but we look at unique markers that make people a little different. So as you can see here, they're similar in one way, but they're different in another. And these genes, we can look at how fast, I mean, I should say these sequences, we can look at how fast these mutation occurs. You can do that in a lab. I mean, you can determine that, that's doable. And then this person, we can see they're related in one way, but not with these others. And this is now a genetic distance. So what I can say is, look, they have two in common, one not. So they're probably closer. But I could also look at the significance of the gene too and say, wow, these are so variable, I can't tell. And then we can focus on this one. So sometimes we can focus on this, or this, depending on what you're looking for. So is that getting confusing so far? Because it better be. Because there's a math and there's statistics behind this. And I was actually that idiot that loves statistics, but not this kind. So what we do is we look at probabilities of two parents, and these parents, just decided to give this DNA, that DNA, whatever, to the parent, to the child, in a thing called a haplotype. You're getting a new term now. Haplotype just means a unique DNA sequence that we're tracking. That's all it means. And you belong to a haplogroup. That means a group of people that share this, whatever. So you belong to your parents' haplogroups because you have some of their haplotype DNA. So what we can see is that mom will pass on, one chromosome or another, and you become a mixture of a family. So we can see that these kids here got different haplotypes from their parents, even though they're family members. And if you were to do a DNA lineage on them, they will show differences in lineage in a way, but still traceable to your parents. Now that sounds kind of weird, but this is a difficult thing to do, guys. And what we do with this is we form a rubric that the companies have. We've tried to find this information out. They won't give it away. They won't give away their genes to look at, and that's okay. That's intellectual property. That's allowed. So what they do is they do a rubric saying, this person matches this way with this person and that way with this person, and we can tell if this is your cousin, this is your uncle, this is your brother, whatever, depending if you're in a normal breeding population, blah, blah, blah. And what we do is we do a mathematical match, and we sometimes pick what's called the nearest ancestor. That means the one that's closer to you, that has a closer match, and we also try to say what's the best genes to look for. So these are all decisions made by the company and saying, how do I figure out your lineage? And they do it with several statistical models and come out with an average. And if everybody understands what the average is usually wrong. It just means it's the middle point, and then from there you get these things called deviations, and that's when the lineage becomes unexplainable or maybe erroneous. But they do good statistics on it, and pretty much for animals we have it worked out. For humans, that might be a different story. So what we do with you is we look at several pieces of information. We look for all these little things called haplotypes, preferably in your Y chromosome or your mitochondria. And we look at overlaps, and that's what these numbers are. How much am I related to the next person? 50% to a sibling, whatever. I have a half sibling here I got in there somewhere, and there's cousins and uncles and unknown people in my family, and you match this all up. And you can get what's called a near lineage tree. It helps to have their DNA. That's the problem. And that's what these services do, is they'll ask you, do you have a relative that can fork out the 99 to $200? We'll give them a 10% discount. So whatever. So this is the traceability. But generally what we do is to get what we call, I'm going to call it ethnic profiling. We look at the Y chromosome and the mitochondria. And why? Because they travel purely from generation to generation without mixing, without matching. They don't get confused, and that's a neat thing. And we're not going to go into the crossing over. So what do? We do what's called a Buccal Swab. That sounds like a movie. Okay, but whatever. So we take some cheek sample. You just scrape it around. First you have to wash your mouth to get rid of the cow and pizza DNA and all that stuff, whatever. And then they clean it. They basically extract the DNA, separate it from animal and other DNA that's in your mouth, because horrible things go into your mouth probably, and whatever. And we clean it, and then we chop it up, and we look for those things I call haplotype segments. That means we chop up the DNA and look for what we want. Little pieces that we statistically found work to follow your family tree at different levels. So what do we mean by different levels? Genographic Project did a beautiful study where they went to indigenous people. We found out my dad was an indigenous Phoenician whose culture went around the world depositing children, which was great because his Y chromosome shows up in a lot of Europe. My mom's family, we're related to actually Mongol-Hungarians. Magyars, the original, we're not the original, we killed off the original, but whatever. Okay, we're the next original. Okay, so whatever. So we found out that they were a Mongolian Turkish race mixture through this because we were able to trace the mitochondria, and they find mitochondria of both groups. In us, it's kind of funny. Okay, sometimes hard to detect. Are you Asian or are you Turkish? Is a funny thing now in Hungary. Okay, so what we do is we collect data from ancestral people that are still living in those countries, and we kind of find out what does your mutations look like, and how old are those mutations, and that's kind of neat. And we develop a database that colors the world, and we're going to look at these migrations a little better. So we can color the world in all sorts of pretty colors and give it labels. And I want to, let me go on to that one though. Okay. So how do we label these haplogroups? We label them by looking back at our ancestry. And what we can do is we can look at original human populations. And this is some assumption. This is also based on African data. We can look at what's called a certain haplotype mutation. Label it L for original person, okay? And then what we can do is say L1, 2, and 3, these people are genetically related from the same lineage, very short distance. And then in that population, we had a big haplotype change. They don't look like L anymore, so we call them what? M. That means they're existing at the same time now in Africa. All these people, they came a little later. They, of course, came a little, maybe at the same time. I mean, their mutations would occur quickly or slowly. Then what happened is people moved out of here, the M's moved out, and there's a whole bunch of reasons why. We think a lot of it was due to early prejudice, or just the fact that people, what happened was a large population moved out and only the M's survived. There's a whole bunch of stories on that, because there's been multiple migrations out, people that we don't even see, haplotypes that died off, but M happened to be successful, whatever reason. M now changes to what? N, and then what, and this is actually called the Eurasian split. So we have the Europeans and the Mongolians, which guys, Asian people and Caucasian people have very close, similar lineage compared to African people, okay? And then what we see is that the N did very successful here, and they had their own haplotype changes over time, from old to new. And the same is true for the Asians. They moved down here. We start looking at more Polynesian populations, and then we start looking at more Northern Asian populations, some of which eventually became what? Native Americans. And notice that the different colors, there's not one type of Native American. They're all different haplotypes that came out of that M lineage, and sometimes even out of the European lineage, we're finding out, when you look at some of the lighter skinned indigenous people versus the darker skin. So this is a map of ancient travels. This is what Genographic Project does for you if you want that. And this is what they told me about my dad, because he's one that they don't have up here. He's a very unique inbreeding group. That's nice to know, whatever, that tend to settle in this area and basically stayed forever and ever and ever, and then later branched out and left their DNA all over basically this region. They were boat people. They traveled by boat. And they even traveled up rivers and ended up in an area called Bessarabia, which is now Belarus, where my dad's family, we know, came from, at least from the immigration papers that weren't fudged. Okay, so guys, this is a chronological map of humans that we assume are ancient, and then modern mutations. So this is the timestamp. So when you get this data back, you will see, you will get letters, usually from mitochondria or from the X chromosome. If you're a male and you donate your DNA, you'll get both, and that gets to be fun, because you get a little more complex map of the female's distribution. Now guys, what's neat about this too, is when you see these changes, these are based on pure populations of people that inbred, hate to tell you that, and that moved around and probably inbred. So this is also called an inbreeding chart. That means these are tribes of people that moved around and bred with their own kind. Is that true today? How many of you don't mate beyond your family? I mean, this is the thing. So we have spread out, and the newer maps are a little confusing because people kind of overlap and whatever. So these haplogroups that you get, the data you get, is basically more of a descendant tree from the Y chromosome and again from the mitochondria. And this gives you a plot of where I fit on these maps. And this is a Y chromosome map. The colors represent, for a particular haplotype, the intensity of where that is found. So what we do when you get your haplotype, we say, my gosh, most of your haplotype is from here. But notice this, this and this are almost the same. We find people in southern Spain and Portugal have almost identical haplotypes to people up here. And what's funny is they look different. That's not due to the Y chromosome or the mitochondria, that's due to your autosomes. And it's also due to people coming in from Scandinavia and raping and pillaging. And also Romans coming through and raping and pillaging. And my mom's family coming in and raping and pillaging. They're what determine, again, some of our appearances, because that's the autosomes. So I can't do this with autosomes, because they're all over the darn place of families coming in. Because I can tell immediately if you're Mongolic, if you have a Mongolic mitochondria or Y. But this is just looking at probabilities of where people came from. That's your ancient ancestry. And we can come up with probabilities in a minute. And this is for the, this one's, I believe, this is the mitochondria from a mother. And what's funny is females show more stable distribution than males. That's because males traveled, males went to war, males did this and that. Very few females traveled. Okay, so we can again look at your mom's family and say, my gosh, this is your haplotype, you probably belong, your family came from here. That's the ancient lineage. And that's easy, but it's still based on probability. We can't even tell what these people look like, even though there's some people now that are doing forensic DNA analysis and using what's called ethnographing, that we can try to predict what a person looked like without even knowing that's their DNA. There's a guy that does that right now. He's an expert on it, supposedly, and he can take DNA from an unknown suspect and build an ethnic profile of this person based on that. Which, how good is that? We don't know, because basically today we're all mutts. So guys, so after people started traveling by boat and getting around by foot long distances and mingling and invading and conquering, guys, you have autosomal DNA from all over the place. And this is what 23andMe also tries to look at. And this is where you get the percentages. My wife was kind of funny because she had hers done first. And this little award I got from 23andMe, they gave me a free kit, which is like, it's just kind of funny running out of time. Okay, I'm gonna take a couple of seconds. Okay, because then I want to have to take time for questions. So with the autosomal, and they do this on dogs actually, you can tell the percentage of breed of dog. So in your autosomal DNA, what we can tell from you basically is who visited. But it also depends on the gene. The problem is there are genes that are conserved. So all of you, if you're Caucasian, you have Asian genes in you because it wasn't distinguishable. So it doesn't mean you're part Asian, it just means you have that common gene. And all of us do to a certain percentage depending on how much we inbred or how much you outbred. Now my wife's came back with a lot of Neanderthal. That's why she's waiting for me to do mine, because my nickname around the house is Neanderthal. Okay, so besides other things. So she's hoping I'm like twice the Neanderthal she is. She's a little upset by that. But guys, we're descendants. We co-evolved with Neanderthals. And we might carry their conserved genes. Now you can look at the recent genes, it means the ones that evolved over time, and we also find those in us. And the genographic people tell me that they think that we mated with Neanderthals and that's how it got in. They're sticking with the story, whatever. Okay, and we also find in Africans that South Africans do not have Neanderthal at all because they didn't encounter them. They lived in Europe. And when we migrated to Europe, all of a sudden Neanderthal started appearing. So if you get that back, don't worry too much. Don't start saying, I'm gonna walk on my knuckles and have a hairy butt. That's not gonna happen. Okay, but what is neat about it is that we know they're in there. And we find other ancient human DNA in Africans of other people they encountered that you wouldn't consider human. And this is kind of neat. So yes, it gives you some curiosity. But as far as what do you learn from this, guys? You learn that you're a pie chart out of this. And you can sit down and guys, look at the world today. And this is looking at haplotypes that are found today throughout the world. And we see that when we look at even simple DNA, that means a true lineage, is that one country doesn't have a pure haplotype. We're pretty much spread out. But we can still tell you by looking at these charts is that if your family comes from this part of the world, there's probability of all of this in your autosomes. Now, it's your Y or your mitochondria that might be a pure this, this, or this. But you're not going to be one pure thing. And you're going to find bits and pieces of information. So a lot of the data you get, just understand, guys, is going to be favoring one lineage, usually, because that's what they pick. They look for the lineage that you show the most familiarity with, which means all of these relatives might be gone. They're not going to be in the picture until eventually you find them. So it favors one lineage. It favors certain genes we're looking for that we correlate with coming from a particular location in the world. But to predict what a person looks like is another story. Now, what these companies, and gosh, we can find every gene in there from you. So if you want to look at a family tree, we find all of those genes in you. But again, it doesn't mean that we belong in the bush with them, okay, in a way. So again, we see all this family lineage. So what is the caveat? What is the warning of doing this? And this is where I think is the most important point, is commercial tests try to do the best match they can. And they try to give you the data based on percentages that are skewed towards one family or another. It can't give you all the information. And most of you, because of the way the world is today, are going to have bits and pieces. But be careful of the probabilities, because a brother or a sister will have a different one. So what you can do if you know your math is you have your whole family do it, and you average out the Celtic genes, the Viking genes, the Mongol genes, and then you could build a lineage. So you yourself can give the information. And I hope this was clear today, because some of this is muddy and muddled. And I'm going to end this with here. Be careful who you go to. Okay, because I've seen this done at the taco trucks already. And also, any quick questions, because I'll stay around.